welcome to our June Salish Sea Roundtable. The idea of this group is an opportunity to learn about topics that have cross-border, interdisciplinary areas of interest. Today's topic is on hatchery effects of trait variations. I am joining you from West Vancouver, which is home of the Squamish, Tsleil-Waututh, and Musqueam traditional territories. I'm located at the Pacific Science Enterprise Center. First Nations used to use this area in particular from harvesting fish in Cypress Creek. So I'm happy to be joining from here today. Dr. Tim Healy will be presenting his topic today. He's a research scientist within DFO, and he's presenting from Nanaimo. As Mariel mentioned, Q&As we'll have time for at the end of the presentation. If folks can turn off their cameras to preserve bandwidth as we go through. And at the end, hands up is certainly a good priority when we're doing the Q&A. We'd like to see smiling faces on the other side of that. Part of this is to connect with others across the border and within. So with that, I'm going to turn it over to Tim, and the presentation should be about 35 minutes, and then we'll have time for questions at the end. So with that, take it away, Tim.
Okay, let's just make sure. Can I just get a check that people can see the slides and which version of the slides we're seeing?
And they look like the correct version.
Excellent. Okay, that's great. So testing ahead of time works. That's fantastic. So, okay, thank you for the introduction, and thank you everybody for showing up today. I know there's a lot going on in the world between summer really kicking into gear, holidays on the horizon, and let's be honest, there's World Cup games that we're all thinking about at some level. And also, thank you very much for the invitation to present today. So I grew up in Vancouver. I've been around the Salish Sea at least somewhat my entire life. It's a phenomenal ecosystem, and I think it's really cool that this forum exists to talk about some of the latest and greatest science that's been going on that is relevant to the ecosystem. And it's just the cherry on top that I get to present some of the work that we've been doing over the last couple of years. For me, I doubt I'm alone. When I think of the Salish Sea, it really does not take very long before Pacific salmon come to mind. And that's just reflects how important these large anadromous fish are ecologically in the ecosystem. And when we think about how people interact with the ecosystem, if anything, that just becomes more true. They're kind of ubiquitous for me when I think about the Salish Sea. Now, despite all of that importance, again, likely won't surprise this audience to hear that if we take a sort of step back and think about Pacific salmon in general, trends tend not to be great in terms of abundances. Pacific salmon populations tend to be declining, or at least at lower abundances than they have been historically. And this chart just illustrates that. So I pulled this data from the Pacific Salmon Foundation State of Salmon report. And what you're looking at is anomalies in spawner abundance across the 5 species of Pacific salmon broken down by region. And the takeaway here is red fish means lower than historic abundances. And there is variation across species and regions, but there's a lot of red on this plot. And not surprisingly, given the importance of salmon, these declines or lower abundances have caused huge concern, and we've seen efforts of all different kinds taken to help mitigate and push back against some of these losses. One of the approaches that has been taken is the production of fish from hatcheries. And this has happened in countries all around the Pacific, but in Canada, hatchery production really falls under the Salmonid Enhancement Program here within Fisheries and Oceans Canada. So SEP produces a little over 300 million juvenile salmon annually that are released into BC waters. Those numbers may seem high to a lot of people, and that's because this includes output from regulated spawning channels. If we talk about hatcheries themselves, most of the production is based around Chinook and Coho, and we're talking about 40 or 8 million juveniles annually, respectively, for those two. Across all of SEP's hatchery production lines, there's a range of different objectives for the different stocks. And we could dig into it if we want, but fundamentally, they're all about supporting vulnerable stocks and harvest, and that is taken word for word from SEP's website. So I'm not making that up. That's what SEP says. Now, hatcheries, as a way of supporting salmon, have some potential. And actually, we can see it in that chart that I started with. These abundances don't separate out hatchery spawners from natural or wild spawners. And you can see that some of the greens, the numbers are really high. The anomalies are elevated. And if we focus on the boxes that I've surrounded in that magenta color, I would argue those are the regions most relevant to the Salish Sea, most likely. And you can see that for Chinook and Coho, they're actually above historic averages if you take it as a regional average. So how can hatcheries do this? Well, fundamentally, what a hatchery is doing is it's adding an artificial component to a habitat for a population, or at least within Canada, that's how our hatcheries run. They're integrated with a natural system. These artificial habitats have vastly higher survival from egg to the stage of release than the corresponding survival in nature. So basically, spawners produce more juveniles per spawner out of the hatchery in that generation. So that's great, right? Seems perfect. Hatcheries will work awesome, except there's a catch, because there's always a catch. And that's that we really can't ignore the fact that it is an artificial habitat. And an artificial habitat produces risks that we need to at least account for as well. This is often kind of conceptualized using a framework somewhat like this. So the idea is that different habitats select for different traits. Optimal traits in one habitat are unlikely to be the optimal traits in another habitat. And that's what's represented in the little figure on the left here. The purple line is showing a trait distribution and the associated performances of that trait distribution in the natural habitat for natural origin fish. The orange line is showing a trait distribution for hatchery origin fish, and the performance is their performance in the hatchery environment. The key observation, though, is those two curves don't overlap very much. And so once hatchery fish with that trait distribution go out in nature, we would expect the performance would be quite low. Now, it's very much so exaggerated for the purposes of demonstration in this figure. The curves essentially don't overlap at all. That's probably not quite right. But the overwhelming majority of studies that have looked at performance differences between hatchery fish and natural fish have generally supported this idea. Hatchery fish tend to perform more poorly in nature than natural fish do. Now, given that, it's perhaps not surprising that DFO and CEP have genetic guidelines to support the operation of their hatcheries to help mitigate against some of these potential negative effects. And I love this story because CEP has had these guidelines since the 1980s. And as it was relayed to me, basically the guidelines came about because a bunch of people got together, had a conversation, wrote them down, and that was that. Which, wouldn't it be great if it was that simple now? But it's not. And there are good reasons for that. And as a part of that, a couple years ago, we reviewed those guidelines, and it was remarkable how consistent guidelines that DFO has had since the 80s were with current best practices. I think across the whole thing, we recommended 2 very minor changes. Great. On top of that, there was a recent advice piece through DFO that took account of that framework I mentioned earlier with 2 different trait distributions and provided advice to CEP on how to account for that for their different hatchery stocks as well. So if you think about what I've told you so far, I've kind of posed a problem, suggested hatcheries are a tool, posed a potential risk, and then told you that we kind of have guidelines to mitigate it and we know what's going on. So why am I still talking? and the answer is it's just not that simple. It's never that simple. There's a lot that goes into what determines trait variation in any species, and salmon is no exception. So for example, there are genetic effects that impact how traits are expressed in organisms. And this is what we've been talking about so far. And it's a little schematic example. So the purple box is supposed to represent a natural habitat. Orange fish are hatchery origin fish, purple fish are natural origin fish. The natural origin fish are bigger, they produce bigger offspring. Hatchery origin fish, smaller offspring. Putative genetic effect, exactly what we've been talking about. But you can also have environmental effects. So now I've added another habitat, I've added a hatchery habitat. And in the hatchery habitat, the hatchery fish are producing smaller offspring. But once they go out in nature, those same fish genetically are now producing larger fish, more similar to what natural origin fish were doing in the first part of this example. And so that's a pure environmental effect, and it could easily be going on as well for a lot of these different traits. And if that's not complicated enough, these two types of effects can interact. So different genetic variation can have different environmental effects. And in this case, we're seeing that natural origin fish do exactly the same thing in hatcheries and natural environments, whereas hatchery fish are showing that pattern that I just described for the environmental effect. And that's a pretty simple interaction. And so the problem is, when we look at trait variation in adults and we haven't controlled for things, it is virtually impossible to know exactly what is causing the trait variation that we're observing. And why this matters for hatcheries is, already told you how hatcheries could generate genetic effects, but I started with their different habitats, and we would predict that hatcheries would produce a whole bunch of environmental effects as well. So what evidence do we have for genetic effects, and particularly genetic effects that come with long-term fitness costs? And that's where the evidence to support a lot of this gets a bit thinner, even though the overwhelming majority do show performance differences. So it's really difficult to assess fitness in a salmon, and most studies that have tried to look at this measure something called relative reproductive success, which is essentially the difference in family size that hatchery and natural origin spawners produce when they spawn in the natural habitat. This has been looked at several times, and by and large, it is a conclusion that hatchery fish have lower relative reproductive success. The average is usually around 0.8, but there are some catches. Oftentimes, these studies still confound genetic and environmental effects. There's a ton of variation across different systems, so it's hard to know how much the average applies in all cases. And the only one study has actually demonstrated that any difference in relative reproductive success actually persists across a generation, which would be consistent with these long-term effects. The reason why it's only one study, or at least only one as far as I'm aware, is that that's even harder to do than assessing fitness in the first place, because you have to go another generation and keep track of everything. So not surprisingly, there's been modeling work to try to bridge that gap. And there are conclusions based on models that there are long-term fitness costs. And in some cases from models, even if you remove the hatchery, the fitness cost is maintained. There's no recovery of fitness, which is a little bit concerning. And the only thing I would point out is, like, those are based on models. Those are not empirical data. So it's hard to know for sure if that's what will happen or how generalizable it is. And interestingly, there was a study from DFO that was published in 2021 looking at Inch Creek coho salmon, which is a heavily hatchery influenced population. And over 3 generations, that study showed declines in traits that are putatively associated with fitness, as we might have predicted. They also showed that those changes were heritable, so there was at least a genetic basis to some of that change. Again, what we would have predicted. But interestingly, their conclusion at the end of the study was that the population still harbored substantial genetic variation and heritability for these traits, which implies that there's the capacity for adaptation, which is a bit different than this idea of irreversible long-term effects. And so there's a lot of unanswered questions. So having said all of that, given it's difficult to study fitness, what can we do? And what we've been doing here as a part of the Pacific Salmon Strategy Initiative is taking advantage of the fact that there are now published genomes for Pacific salmon and using 2 approaches to dig into some of these questions. The first is to take a naive approach. We just screen the entire genome of Pacific salmon, look for differences between hatchery and natural origin fish, generate hypotheses based on those differences, and then test for functional differences, all to build support for fitness effects. The second approach is to sort of do the opposite, where we predict a trait that we think is tied to fitness, and then look for genetic associations with the trait variation, and then look at how hatcheries might impact that genetic variation. So I'm going to go through an example of each of these. Predictably, I'll start with the first one, and I'll actually start with just the first bit of trying to identify variation between hatchery and natural origin fish. Because believe it or not, it is a valid question to ask, is there genetic differentiation at all, particularly in integrated systems? Because low density marker studies from the past have suggested there isn't. So what we did was we took 4 different hatchery stocks of Chinook salmon from the west coast of the island. We identified origin of the returns using parentage-based tagging. And then we used pool-seq to look at genetic variation across the entire genome. Briefly, that's when you take a lot of individuals, pull them together, sequence them at high depth, and the reads to each genetic variant let you estimate an allele frequency difference between different groups. So with all that said, what did we find? Turns out low density marker studies more or less captured the pattern. So we saw very little evidence for genetic differentiation between hatchery and natural origin fish. What I'm showing here is a plot of fixation index on the y-axis across the Chinook salmon genome for Chinook or for Sarita River Chinook salmon. The alternating grays are different chromosomes. And what fixation index is essentially a measure of the proportion of genetic variation that occurs between 2 groups. And it ranges from 0 to 1. 1 is completely different. 0 is the variation is spread evenly among all the fish that you tested. And you can see our averages are hovering right around 0 across the whole genome, and the maximums are only getting up to around about 0.1. So we see almost no evidence of genetic differentiation between origins, which is a bit interesting if you think about it, because you can measure the performance differences within the same generation, but we can't detect genetic differences. Now, you could argue that's because integrated systems are managed to hit equilibrium, but it's still a bit of a logical disconnect. It's possible that's because these traits are all polygenic and we're looking at subtle allele frequency differences. And it's possible that this is consistent with weak selection, but we're certainly not seeing like a big signal that you might predict if there's these big irreversible changes. And just so you're convinced I'm not cherry picking this result, all 4 systems look almost exactly the same way. There's a little more going on in NitNat, but we're still talking about 0.1 as a high point, absolute max. So that's a bit puzzling. And it poses the question like, well, is there something else going on? And there's an alternative possibility. And that alternative possibility is something called epigenetics. So epigenetics literally translates to on DNA. And these are a class of modifications of DNA structure or organization that have the potential to be inherited. So genetics is change in DNA sequence. Epigenetics is change in structure or organization. There's a whole bunch of different types of epigenetic modifications, but the one that has been studied and characterized the best by far is something called DNA methylation. And without trying to bore everyone with biochemistry, DNA methylation is the addition of a methyl side group onto a specific base pair in the DNA. It forms a covalent bond, so it should be stable unless an enzyme comes along and changes it. And these modifications can affect function through changes in gene expression and regulation. But we have a potential mechanism that could be inherited that might explain differences between hatchery and natural origin fish. And I didn't come to this randomly. This really hit the scene in about 2017 when a group published this study on Capilano and Quinsam Coho that showed that if you look at hatchery fish in blue or natural fish in orange, you can see that there are differences in methylation if you look at over the whole genome. So the plot is showing individual variation in DNA methylation in semantic space. And what that means is points that are closer together are more similar, points that are farther apart are more different. And what you can see is that the orange points are spreading out towards the bottom right of the plot away from everything else. And that is a significant effect consistent with an effect of origin or rearing environment, as they termed it. And this is all data from white mussel. This study concluded that there were consistent effects across hatcheries and that they were substantially larger than genetic differences within the same stocks, which is really promising and interesting, but it poses 2 questions. First of all, that's coho. What's going on with Chinook? Because that's the bulk of the hatchery production in Canada. And second, that's only 2 hatcheries. Are we really looking at a consistent effect of origin, or is it just that these 2 hatcheries have some similarities? So what we did was we took samples from 8 different Chinook salmon hatchery stocks. We used fin tissue. We determined origin by parentage-based tagging. You can see where the stocks are on the map, and there's a bunch that are relevant for the Salish Sea. We took those samples and we did whole genome methylation sequencing, which is a technique that lets you measure DNA methylation across the whole genome. And then we compared patterns between hatcheries and origins. What did that look like? So here I'm showing our data in the same kind of analysis. This is a semantic space plot. So the points are individuals in the data set. Closer they are, the more similar they are. The colors indicate the different stocks, and the ellipses indicate the 95% confidence intervals for the distributions of the stocks in the space. And there's a couple takeaways here. The first is the ellipses don't all overlap. So stocks have, maybe not unique, but stocks have different methylation patterns depending on where you look. So it's just like genetics, you can tell stocks apart with DNA methylation. The other thing that jumps out here is that there are these two obvious clusters that I pointed at with the two reddish arrows. And those looked really funny the first time we saw them. That was not expected. And it turns out there's a good explanation. So the samples we were using are all part of our broodstock monitoring program, and different hatcheries prefer different fins when they take their clips. And so what you're looking at there is the difference in DNA methylation between an adipose fin and a caudal fin. It is a massive difference. And it's worth highlighting because unlike genetic differences, DNA methylation is tissue specific, and it can respond to the environment because it's an enzymatic process. So it has the potential to be inherited and be like a genetic effect, but it also has the potential to contribute to environmental type effects or tissue type effects. And so it sits right in a sort of intersection between everything I've been talking about so far as a potential mechanism. Leaving that aside, now that we know that the fins are really different, what about origin if we separate the data set by fin type and look at origin effects? So again, semantic space plots, and I've recolored it. So hatchery origin fish are orange, natural origin fish are purple. The ellipses are the confidence intervals again. And regardless of whether you look at adipose tissue or caudal tissue, those ellipses overlap. There is potentially a little, maybe something going on in caudal fin for about 8 fish, but that's it. So we do not see any of that same overall signature of origin or differences between hatchery and natural origin fish in the patterns of DNA methylation across the genome in our Chinook salmon data set. Again, bit of a surprise. The obvious follow-up is, well, we don't see anything consistent, but what about if you look at individual stocks separately? And that's what I'm showing here. So this is a different type of plot. This is a heat map. So what you're looking at is each column represents an individual. Each row represents a region of the genome. The more blue shading, the higher the DNA methylation in that region for the individual. The orange bar on the top shows you the hatchery origin fish on the left. The purple bar shows you the natural origin fish on the right. Bottom line, for Chicomis River Chinook salmon, there's a little over 100 regions in the genome that are differentially methylated between hatchery and natural origin fish. They're spread throughout the genome. There's no like peak of this is where it is, but there are differences. And I'm not going to take the time to go through this figure for all of them, but basically it would look exactly the same. We're talking a small number of regions across the genome that are differentially methylated by origin. And that's summarized with this little figure here. So the diagonal gray bars show you the number of regions that are differentially methylated between the origins for each of the 8 systems. The reason why I'm showing it like this is that the white boxes underneath show you the pairwise comparisons between systems, and you can see that almost all of those numbers are 0. Even when we look at directly comparing regions that we detected, there is almost no evidence of a consistent signature across these different systems. So if we come back to the questions I started with, are there differences in Chinook salmon? Yeah, there are. You can detect them. They're there for sure. It's a bit of a debate whether there's more or less genetic signature, but they're there. But there's no evidence of a consistent effect. And my colleague Kyle Wellband went back and reanalyzed some of the coho data and some new coho data, and that conclusion holds for coho as well. We do not see any evidence of a consistent effect. But we do see effects in each system. And I already told you that DNA methylation has the potential to be inherited, but it also can reflect reversible patterns. So there's an obvious follow-up, and that's, is there evidence that any of this is inherited in the next generation? And I don't want to go into it in depth, but Kyle's group did some work on that. They used Chehalis River coho salmon. They compared fish of the different origins. They brought them back to the lab. They did controlled crosses. And if you look on the left side of each panel are showing adults of hatchery origin in yellow, natural origin in blue. There's a difference in methylation. And then if you look at the offspring, the yellow and blue ones are the offspring of pure crosses of those types. And you can see that they mostly track the parental patterns, or at least they're closer to their parental patterns than they are the other parental. And in the middle, you have the two hybrid crosses. They're kind of in the middle, but it depends on the region you're looking at and exactly the mode of inheritance that it looks like. But regardless, yeah, it looks like, at least for some candidate regions, these patterns do get inherited across generations. So we've got some differences now. Next step is arguably the hard stuff, and that's generating the hypotheses and testing the functional effects. That work is ongoing through a collaborative grant with funded by Genome BC with researchers at UBC, Patricia Schulte and David Metzger. And so we're excited to see where that goes. But for the purposes of today, let's move on to approach 2. And let's turn the whole thing around and start with a trait that we think is potentially linked to fitness and look at genetic associations with that trait. Step 1, let's pick a trait. And we're going to pick variation in seawater residency time. And we'll explain why in a second. But Chinook salmon show a huge range of life history across their range and even within populations. In particular, the amount of time they spend in the ocean can vary quite widely, and it has a big impact on the size that the fish return at for spawning. Not surprisingly, older returns tend to be larger. Why does this matter? Well, there's the simple statements like people like big fish, orcas like big fish, salmon like big fish. But to be a little more technical about it, salmon like big fish because large size is generally a competitive advantage in spawning. For males, that's through mate competition. For females, it's directly through higher fecundity and competition for red sites. That then poses the question, well, why aren't all salmon big then? Because if big is good, why? And it's most likely that those benefits are offset by the extra time in the ocean and the risks of mortality and predation that come with that. Because remember, if a salmon doesn't get back to its natal spawning grounds, fitness is 0. We know that. It's one of the easiest things we can measure for fitness. And so generally, large is good, but it's a balance with risks in the ocean. Now, the really interesting part is that males have an alternative way to bypass all of this. And that's that there's an alternative life history strategy in males known as jacking, where males return 1 year earlier than the earliest returning female in a population. They return smaller. I boxed the 2-year-olds in this photo because those are the jacks for this system. And those much smaller males participate in spawning through a sneaker male strategy. So they're not directly engaging in competition with the big males for females, but they're sneaking in when spawning activity is happening and contributing to the next generation that way. And it's been shown that this trait is at least putatively under negative frequency dependent selection, which is a fancy way of saying that if there's a lot of big males, being a jack can be a good thing. Whereas if there's a lot of jacks, it's generally better to be a big male. Regardless, though, the main point here is even if jacks overall have lower fitness than bigger males, they have higher fitness than all the males that stayed out in the ocean after age 2 that died. And that's where you can see how this trait might persist. Additionally, there's been a bunch of studies that have shown that this trait has relatively high heritability, which implies that there's a genetic basis for this trait. Why would we pick this trait if we were thinking about hatchery effects? Well, hatcheries bypass spawning dynamics. And so now jacks are no longer at a competitive disadvantage unless the hatchery puts them at a competitive disadvantage. So a hatchery could enrich the population for the genetics that underlie jacking in males, which would then push the whole population to smaller sizes and younger ages, which is generally not desirable. On top of that, there's a lot of evidence that suggests environmental effects early in life affect whether or not males come back at younger ages, and that's exactly where hatcheries have their effects. So a hatchery could impact this trait both from a genetic view and from an environmental view. Now, when we were thinking about this, thankfully, we didn't have to start from scratch because folks up in Alaska and at NOAA had already thought about this. And they posed a very logical question of like, it's a male-specific trait. Wouldn't it make sense if it was attached to the male-specific chromosome? And so what they had already done was characterize variation on the putative Y chromosome or male sex chromosome in Chinook salmon up in Alaska. And they characterized 4 different versions of that putative Y chromosome and showed that the different versions not only affected the age of return, but also the size of the males in the year that they returned. And that's what's shown on the right, which is super interesting. Some of the same researchers then did arguably a more detailed study in the Columbia River, looking at variation age of return in a single population. And I'm summarizing that here. So on the left, you're looking at a genotype plot. So now each column represents a site in the genome, or on the putative Y chromosome in this case. And the red and yellow colors are different genetic variants. Each row is a version of the chromosome. And the vertical bar on the left that are red and blue is showing males and females. Males are in blue. The takeaway here is that there's this consistent repeated banding patterns at the bottom of the plot. And almost all the individuals that have those banding patterns are males. So these are male-specific haplotypes, high linkage, consistent with putative Y chromosomes. And what they showed in this study was that if males had haplotype A, regardless of if they were hatchery or natural origin, they were most likely to come back as jacks, which are age 3s in this group. Whereas if they had haplotype B, natural origin fish tended to come back later. Hatchery origin fish, it was more distributed across all the possible ages of return. We're likely looking at environmental effects on top of the underlying genetic effect. What was interesting is that the haplotypes were different. If you looked at Alaska and here, which implies that there's some population specificity here. And that led us to the question of, well, what's going on in Canada? Because obviously that's our focus at Fisheries and Oceans. So what we did was we basically took the same kind of approach that was used for that Columbia study, and we collected genetic variation for 4 different stocks of Chinook salmon from hatcheries in southern British Columbia up into the central coast for Narco. And we used a reduced representation sequencing approach to characterize that SNP variation. The sample sizes are shown on the plot. And again, we determined the age of all of these samples using parentage-based tagging, which was possible for these populations. This plot shows how the samples parse out by age. The details are not important here, but the thing that is that for at Narco, females tend to come back as age 4 and 5, which would put jacks at age 3 if we stick to the definition I presented earlier. Whereas for the other 3 systems, females come back at age 3 and 4, so jacks are age 2. What did we get for genetic variation among these different groups? This is another one of those semantic plots looking at now genetic variation. We had about 56,000 SNPs, or single nucleotide polymorphisms, characterized in the study. The Narco fish and the Chilliwack fish were distinct from all the others. Puntledge and Qualicum are that cluster on the bottom right that fall on top of each other. There's a history of transplanting. That wasn't a huge surprise. And what we decided to do was analyze that cluster as a single unit, which because the genetics were similar. Step 1 was to say, what's the sex chromosome in these populations? Because we have a guess, but there is some variation and we don't know until we check. So we did a genome-wide association study for differences between males and females, and we'll play the Sesame Street game. One of these things is not like the others. There's a huge peak right on OTS 17, which is the chromosome we would have predicted. That's the sex chromosome. So now we did the exact same approach that they took in that Columbia study. So how that works is you take the variation on OTS 17, you subset down to the highly linked loci, and then you phase that into versions of the chromosome, and you generate these plots. Same type of plot as before. And you can see that we capture these sort of repeated banding patterns. They're a little different in how they appear, but they're still there. And if you look very carefully on the left, 4 of those clusters are essentially only found in males. And if we pretend those are Y chromosomes and call genotypes, that's summarized in the table. And you can see we do a pretty good job of coming up with X and Y genotypes for males using these 4 haplotypes for Narco River Chinook. If we take those haplotypes and look at ages, this is where it gets strange. So there are significant differences. That's step 1, but it's not what was seen before. So if you look at jacks on the left, the obvious difference is that there is no occurrence of that second haplotype that's sort of a dark teal color in jacks. And then it appears in age 4s, and it's the most common haplotype in age 5s. And the other 3 haplotypes are essentially the same between jacks and age 4s, and then they're at lower frequencies in age 5s. So we do see significant differences with these haplotypes in age of return, but it's not that there's a haplotype that's highly predictive of jacking. Now, just quickly, if we look at the other systems, do the same thing in Chinooks, you get 2 haplotypes. Again, we do a pretty good job of getting X and Y genotypes. Puntledge qualicum, same story, 2 haplotypes, pretty good job of X and Y genotypes. Now, if we look at how those haplotypes distribute with the ages of returns in those systems, there's no effect at all. Jacks, older males, regardless of age class, they all look exactly the same. So we did detect effects, but there are differences before, and it's not consistent across populations. And so some preliminary conclusions, like what I just said, we do see effects. They seem to be population specific. They seem to vary a lot. We don't have haplotypes that are highly predictive of jacking, although it's important to note these are all hatchery fish, so environmental effects might be masking things, and that would be interesting to figure out. But the one thing we can say is, if SEP and hatchery production has caused an increase in these younger age classes of males in these populations, we don't see it as a big difference in the Y chromosome haplotype frequencies. And so that suggests that if anything, it's pointing at an environmental effect. And if you look across the whole genome, that holds. So having said a lot, what can I leave you with as a takeaway? I want to be very clear here. I am not in any way saying that there are no genetic effects of hatcheries. I am not suggesting that there are no genetic risks associated with those effects. And I'm not suggesting that a precautionary approach isn't the right thing to do. I think all of those things make a lot of sense. What I am trying to point out is that the basis of trait variation is really complicated, and there is a lot that we still don't understand about hatcheries' impact salmon. And whether you think about hatcheries from a point of like maximizing efficiency of production and harvest opportunities, rebuilding populations, or mitigating risks to wild populations, understanding these effects is critical because it determines what you would predict for long-term consequences and risks. And with that, I'll just acknowledge and thank all the folks here in our molecular genetics section that contributed to the data I presented and just putting together some of these ideas, our academic collaborators at UBC, and our various sources of funding. And hopefully I left at least a little bit of time for questions.
Oh, that's great, Tim. Perfect. You're right on time. I just was showing my smiling face just to know that we're getting closer to our completion. So I think the timing was great. Loud round of applause for all. So for Q&As, you can raise your hand and we will unmute you, or you can type it in the chat and I can read it out. That was a lot to digest. Tim, you always have a very dense presentation and reminds me that I have not done enough genetics stuff lately to be, but you're also able to translate that information into folks that are not working in that sphere regularly. So with that, we'll open it up to questions, and kind of interesting to maybe even get some of the perspective from across the border for any hatchery folks down on that side of the border of work that's going on down there, and how this could be applicable, or similarities or differences or anything that they found too. So with that, I'll just open it up to questions. Usually takes a minute for people to kind of So, and I'm going to butcher some names. I think we can turn on the hand up function. Someone's looking for it.
Working on it, but Israel is unmuted.
Oh,
Okay.
thank you. Yeah, my name's Israel.
Go ahead.
My name's Israel Payton. I'm Director of Sport Fish for Alaska Department of Fish and Game. Great presentation, Tim. I'm not a geneticist or a scientist, so a lot of it was, pretty complex. One thing I would like to go back to on questioning, like your age and your jacking. So that's what we're struggling with here as far as nomenclature. And I was trying to follow along, and your ages confused me a little bit. In Alaska, we refer to them as 1-1s, 1-2s, or 1-3, like a salt age. And then the overall age, like for a 1-3, would be a 5-year-old, is what we call it, because we add in the 2 seasons basically. So, and we call jacks the smallest returning fish. So we call jacks like the 1-1s. Granted, most of the all 1-2s, I think 95% of the 1-2s, and I think maybe that's what you're calling 3-year-olds, are male. Like we'll get a few, and usually they're under 28 inches. But can you walk me through your ages? Because you were referring to
Yeah.
3-year-olds a lot. And in my
Yep.
world, that would be a 1-1, which would be like a 20-inch fish. But
Yeah. It is very understandable that that was confusing, especially in a flyby, because when I first started doing this, I tried to figure it out because in my head I was like, "Oh, a jack, that's one thing, right? This is easy." And it turns out what gets called a jack varies an awful lot depending
yeah,
on the system you're looking at. And like the Columbia gives me headaches because you've got mini jacks and micro jacks, and trying to figure out how that relates to anything anywhere else is difficult. Um, so for this presentation and for how we've been thinking about the data, we used that scientific definition that I posed at the start, which is from, oh, I can't remember which paper it's from, but it really is basing the definition of a jack based on the age distribution in females, with the idea being that if we're thinking about the competitive dynamics, like a male that comes back 1 year earlier and then is participating in spawning and all of that, it's kind of a similar relative effect to the female distribution across systems, but it does create this variation across the board and things being different. So in the systems we looked at, and in the Columbia as well, by that definition, age 3 would be termed a jack. At Narco, age 3 would be termed a jack, and Chilliwack and Qualicum and Puntledge the age 2s would be jacks, which were, and for the ocean type Chinook, those, I think the 2-year-olds would be equivalent to like a 0-2 using the same numbers, but that's where my knowledge of how you term salmon age distributions and label them starts to falter
okay,
a little
gotcha.
bit.
And you might have some natural wild zero checks, where in Alaska most of ours aren't, so that's why we had 2 years. So that might be some
Yeah,
differences too. But one more quick
yeah.
follow-up, and I'll let everyone else. So in all your studies and on the Chinook and those hatcheries we used, I'm assuming all those are first generation backstocking or F1s, P-knob of 1, or are they not? Because when we refer to
They
hatchery fish, there's a big difference between a domesticated hatchery brood fish and a first
yeah.
generation natural backstock one. So can you shed some light
Yep.
on that? Yeah.
I definitely can. So all of the hatcheries in Canada are run as integrated hatcheries. So we do not have effectively any history of like a segregated brood stock where there would be that independent domestication signal in the brood. There have been a couple captive broods for extreme conservation circumstances, but that's a bit different. And so it's not first generation. It's an integrated system where the spawners in the hatchery are a mix of hatchery origin fish that came back to the hatchery and natural origin fish that were either collected for spawning or also came back to the hatchery. The PNOB, PNI type numbers vary quite a bit across different systems. For the ones I showed here, for the most part, you're looking at heavily hatchery influenced systems, but Sarita's more moderate, and a couple of the other ones verge almost to the relatively light hatchery influence. So there's a bit of a range.
Okay, that's interesting. So we have 2 integrated Chinook programs in South Central Alaska, and we keep the P-knob at 1. So we only backstock clipped. We only use wild or natural origin as the brood to backstock it. So that's, which I know that can, there can be differences in fitness and all that when you get into that. So we we keep ours at 1 or 100% wild back stocking. So, all right. So
Yeah, and
yours
I'm gonna jump in
variation of them. Okay.
I'm going to jump in there because we have lots of other hands up and great questions. Israel, that was a great question. You can certainly follow up with Tim after. I've got Gary Marishima with a question. Did you examine the effects of hatchery genetic management plans? And then we'll go to Christopher and Ian. So if you can answer Gary's question.
So with our data sets, although they are, it's hard for me to call them small because they're sort of ridiculously large for this kind of study in some cases. But I don't think we have enough representation of different genetic management strategies to properly test the effect. All we can conclude is that there are system-specific patterns, which likely have, like, there's almost certainly some element of the hatchery management plans and all of that. The other thing is the management plans, at least in terms of genetic risks, all build from the same guidelines, but how those guidelines are exactly applied do vary a little bit, and they can vary through time as well. So it would be great to look at, but I don't think we have the metadata or amount of data that would be necessary to really tease it apart. And I think there was another question after that, Cher. Sorry, what was the next one?
Yeah, so Christopher Warner's question was, do the trends in haplotypes hold true when we examine segregated versus integrated hatchery populations? I think you might have answered that a little bit, that we don't have true segregated hatchery populations.
Yeah, it's not relevant. It's not relevant in Canada. I'm pretty sure the Colombia system was integrated as well, but I'm pretty confident based on a name I saw on the call, there's another person on this call that would be better to answer that than me. And I don't know if there was a conclusion on that for the Alaskan haplotype study or not.
Thanks. I'll put the next question over to Ian Gorter. I think you can, yeah, perfect. Here you go.
Okay, thanks. Yeah. Thank you. Tim, I thought that was a great presentation, and you had me guessing. As a scientist, I'm thinking like, hey, where's he going with this? And you did a nice job of keeping me on my toes. It was hard to sort of pin you down in terms of where you were heading with your research there. So this is kind of more just high-level basic questions. Some of the other folks are more down in the weeds than I am, but I think the Ford 2002 concept has been it's a compelling, simple idea of how it might work. And which is why when you put that up initially, I was like, okay, here we go. It's going to be all about genetics. I thought you did a nice job of characterizing the potential for the environment to play a role in differences in performance of hatchery and wild fish in the wild. And so just making sure I understand your takeaway here. I like what you're doing with looking at genetic and epigenetic issues and kind of unpacking those to try to find evidence for truly a genetic effect versus a behavioral or environmental effect of hatchery and wild fish in the wild. But are you still kind of on a mission there to figure out if there is compelling evidence for genetics being the driver behind fitness differences that have been documented between the two types of fish in the wild? Or are you leaning one way or another more towards environmental effects or genetic effects? Because I wasn't clear at the very end of your presentation there.
Yeah, that's fair. I mean, there was an element of walking a line there, but to some extent, that actually reflects my thinking on it, because the sort of theory behind the genetic effects, everything it has built, the PNI tool, the Ford model, like there's a ton of utility there. But it leaves me with this puzzle to some extent of just like, well, if that's how it works, why is it not simple to see where the effects are coming from? Because it logically, it just feels like that should happen to me, at least more so than it does, I guess. And so if I'm thinking about my background's in comparative physiology, so I've spent the last 15 to 20 years of my life thinking about how genetics and environment interact to form trait variation. So I don't think I can nail myself down to I care about one thing or the other more. What I care about is we're seeing trait differences. If we want to understand the long-term consequences of that or how best to mitigate or manage those effects, we need to understand where they're coming from. And so there's an element of what's going on genetically, what's heritable, all of that for monitoring purposes and everything. But we can't ignore the environmental effects that are impacting everything that we see outside of controlled studies. So I sort of answered your question by not answering your question, but that also reflects reality.
A perfect
appreciate that.
answer, Tim. Thanks, Ian. So Tom's posted a significant thesis in the chat. I'm going to give him the opportunity to ask the specific question that he wants addressed out of that. If he wants to, Tom Chance, if you want to have your hand up, we can give you the floor.
I mean, I won't be able to say it as well as I wrote it. Not that I wrote it very well, but
you were just going to make me read it, which I wasn't very good at that either. So over to you.
Yeah, the genetic theory aspect of all of this, and I manage hatcheries, is very interesting to me, and there are a lot of parallels and inconsistencies across scientific disciplines. But one thing that I've been thinking about is genetic determinism, and which is it's a theory that basically it assumes that genes and genes alone determine and dictate everything about an individual, whatever it may be that has a genotype, and basically that's their essence. So in terms of hatchery origin spawners, whether they're spawning on their own in the river or mating with natural origin fish, lots of people are assuming that the naturalness and their success is predicated on their genetic quality, and that really is a purity thing. You start getting into some concerning areas there. So I think there's a big conflicting assumption here when it comes to evolution. So if those deleterious maladapted genes can be added into a population, shouldn't there be a mechanism to also remove them instead of them persisting permanently, and heritably, like Iraqi and others have concluded. And as a side note, I always like to point it out: genetic determinism is the basis of eugenics, and unfortunately, it's been inserted into the salmonid science world.
Yeah, so I think you brought up a lot of good points and interesting topics tied to the situation. I personally, I think it's not dangerous is too harsh, but I would not go with like genes determine everything. I think there's a lot of very solid evidence to suggest that environmental effects, all of those reversible, irreversible effects in development have a big impact, right? So there's a range of different inputs that all determine trait variation. That's where I would start with thinking about that. But the last thing you said really ties to where I started all of this, and that's that there's this idea out there that if a hatchery has an effect and it's genetic, even if you take the hatchery away, that negative effect is locked in and there's nothing that can be done about it, basically. And I would at least suggest that that's not necessarily how evolution works, and that if there is still variation underlying the relevant traits in the population, it can respond to selection. Now, that doesn't necessarily mean you go back to exactly where you started, but it doesn't mean the population is stuck either. And certainly that study that I popped up from Bob Devlin's group that looked at Inch Creek Coho, that suggests there's still a lot of genetic variation for traits that we think matter even after intense hatchery inputs into a system. So there's a lot still to be done. I'll end the answer with exactly what I ended the talk with. There's a lot still to figure out.
Yes, there is. Thank you.
Thanks, Tom. Appreciate that. Okay, we've only got a few minutes left, so I'm going to just jump to one of the questions on here that was sort of a different topic area. Shauna Hurst, you're welcome to put up your hand too, but There's a little bit of confusion on that. The epigenetic study found that the hatchery and natural origin had no difference, but then they noticed that she identified the differences in the methylation. Can you give a little bit of an explanation about that?
Yeah, sorry, that's my fault. I did not do a good job of setting up those two different slides. So the first slide where I showed a comparison of methylation for hatchery and natural origin fish, I'm not going to try to go back to it for time, but in that semantic space, those big clouds of points with the ellipses on it, that is showing the two dominant axes of variation in the data set. It is a way of summarizing an awful lot of data simply so that we can visualize it and think about it. So what that shows you is that there is not this massive overarching signal across the genome that you see regardless of system that separates hatchery and natural origin fish. The second thing I showed, where we were looking at that heat map and I claimed there were differences, what happened there was took a system out from all the others and did a different type of statistical test to specifically ask what's different between hatchery and natural origin fish in this system. And when you do that, you detect differences. They are just not enough to drive an overall pattern that you would see in the first analysis. Then the follow-up was we did that in each system separately, found differences in all of them, but even when we did that, there wasn't a lot that was shared across any of them. So which then goes back and is consistent with the first observation of there isn't that big overall pattern or a consistent pattern across all the samples.
Perfect. Thanks, Tim. Shauna, I hope that got you the answer some clarity that you were looking for. I'll go with one last question that's been typed in the chat as well. Elise, you're welcome to put up your hand, or I can just read out what's in here. I'll give you a second to make your choice here. I'm looking at too many. Yeah, perfect. Elise, your hand is up. I think Marielle can unmute you. And so this will be our final question. Jump on in.
Cool. Can you hear me okay? I don't have
no problem.
Great. So there's this paper that I read a while ago by Dan et al. I have to go back and have it in front of me. But it basically showed they were comparing natural origin crosses with hatchery origin crosses, and then F1 generation that hatchery origin had spawned in the wild, and the relative reproductive success of those crosses. And they showed that with one generation, so hatchery origin that then spawned in the wild, so that F2 generation had basically no difference in relative reproductive success for all the natural origin fish. Sorry. Works out that explaining as well, but I believe it's an organ system, and I was just really curious. And you kind of answered this when you talked a little bit with Tom, but just some specific. And I think you had some system specific results as well. So just your thoughts.
Thanks, Lisa. You were a little bit echoey, so hopefully Tim was able to get the basis of that.
I think so. I think the basis was describing another study that had looked at these types of effects and the same kind of themes coming through, where there's a lot of system specificity, which without data I would suggest is probably some combination of the genetics of the specific system and the history of stocking practices that were relevant for that system. And I really do think that is the common theme that comes through here. There's a lot of really great work and management actions that are based on sort of average conclusions across the board, that precautionary approach. But as with so many things, I say this in almost every talk I give, and I tried not to, the devil is in the details. And I do think that is relevant, especially when we start talking about Pacific salmon across such a large geographic region. So yes is the answer. And I believe, I don't know if Kyle's on the call, but I'm pretty sure Kyle Wellband is a co-author on a review that came out recently on hatchery effects that essentially puts forward this idea that there is a lot that we don't understand. There is a lot of system specificity, and we still need to work that in. Without abandoning the precautionary approach overall, because there are good reasons to have that in place.
Thanks, Tim. I appreciate that. So it is 1:30. So with that, we're going to wrap it up. There will not be another session until September, I believe, Marielle.
Correct.
Yeah, summer hiatus, right? Yeah, enjoy your field season, holiday season, etc. But appreciate all the folks that joined in today. Tim, excellent presentation, great questions from the audience, and appreciate Ariel for hosting all the online back of the scenes, which we are barely capable of managing internally here. So thank you for that. Much appreciated. And looking forward to seeing everyone again in the fall.